All right. Welcome to chapter 25. Now we're going to be talking about animals. This is our last unit that we're going to cover, and we're going to spend some time on it. It's quite diverse, so we will be looking at trying to look at different things that make animals different. Why certain animals function certain ways, and why is it that all of God's creation is made individually, why we are who we are. So we'll go through chapters 25, 26, a little bit of 26, 27, 28, and 29. So it's quite a bit of chapters. And like I said, there's not a lot of there's not a great way of going about doing all of this at once, trying to reach all 16 million different species of animals alone. So this is all talking in generalities. For the most part, we're going to break it down first, what is an animal, and then we're going to look into different systems and looking at the basics. Now, when I say that, I have to say that because a lot of animals, for the most part, certain types of animals are going to do it within the same way, but you may have one that does it a little bit differently. An example, I'll use plants. What I mean by that is all plants make their own food through photosynthesis. But there are some plants that eat other organisms. So that's kind of that exception. For most part, we're going to talk generalities, but there are going to be some exceptions when we are talking about some of these animals. So we will cover chapter 25, what are animals? So what makes an animal is going to be that of first off, it's going to be a eukaryote. If you look there, going back to the cells that we talked about, they're going to have eukaryotic cells. They're going to be part of the kingdom. So looking at the kingdom, we're looking at all the different taxa. They're going to be part of Animalia, eukaryotic, multicellular. Most of the animals will have very different or differentiated cells, but they have to have multiple cells. An amoeba, an example, is not an animal. Okay, it's a single cell. Okay, so it's going to have multiple cells, and also a big difference between animals and plants is that we are heterotrophic, meaning that we have to consume other organisms in order for us to gain energy, and that we lack a cell wall as well. Now, the big classifications of them, you have really two types of subcategories of animals, and then from that you're going to branch off from those for the most part. Now, the first type is an invertebrate. Now, in being not vertebrate, being vertebrate. So these are your non-vertebrates, okay, or they lack a backbone. And then others are going to be your chordates. Those are going to have backbones, and along those backbones are going to have a dorsal hollow nerve cord, a notochord, a tail that extends past the anus, and pharyngeal pouches. Now, if you're looking at this and you know, okay, well, we have a backbone as humans, but what are all of these? Well, we're going to get into that. Okay, notochord being the spine, or the spinal cord, the dorsal hollow nerve cord being that's pretty much your vertebrae, your spine, the bony part, then the spinal cord being in there. Humans have a tailbone. Okay, it's just fused with the pelvis. And then pharyngeal pouches, that may sound foreign to you. That's what we call lungs. Okay, so pretty much a pharyngeal pouch is just a sac-like structure that's going to exchange gas. Okay, so then, okay, so now that we understand what an animal is, now we have to take a little bit into what do they do. Okay, remember, we want to apply all our life characteristics to what we're talking about. So survival obviously is what needs to happen in order for you to survive. So these things, okay, all those life characteristic things that we talked about early in the year. Okay, the biggest one is maintaining homeostasis. Okay, I'll tell you right now, there's only one way to die. Okay, a lot of people will say that, whatever, a heart attack or a car accident or whatever it may be, but there's only one way to die, and that is to no longer be able to maintain homeostasis. So if you take my forensic biology class, we talk about death, and there's really three things you look at in death. You look at the manner, okay? You look at the cause of death, and then you look at the mechanism of death. The mechanism is somewhere related to here. Okay, that's what leads up to the person not being able to survive. Okay, and your body or animals have feedback systems, usually in the form of some sort of nervous system. What they do is they're going to monitor right here. Okay, so they're going to be able to gather and process stimulus. Whether then, once they gather and process that, then they're going to be able to respond to that of some sort. And they create a feedback system. So anytime you hear the term feedback, you're getting some sort of something from what it is that you are watching or monitoring or whatever it may be. So in your body, you're constantly getting feedback systems or stimulus from internal and external environment. So not only what's in your body, but also what's outside of your body as well. Okay, and if you've heard of this before, the fight or flight feedback system, that has a lot to do with survival, especially in the wild, whether you're going to stand and fight or whether you're going to run. Okay. Also, you got to be able to exchange gas. Okay, oxygen. Got to have oxygen in order to create energy, so they're going to be able to exchange the gas through those pharyngeal pouches, some through skin, some just through cells alone, and so forth. And then also collect and eliminate waste. Now, all of these, I'm not going to spend a lot of time into these. Probably already spent too much time than I wanted. But a lot of these we're going to go into certain systems, okay, and talk a little bit more about these systems. And then so that way you understand a little bit more about how they work. And then finally, being able to reproduce. That's one life characteristics. Being alive, you're able to reproduce. So you got different ones here for you. The first one being, this is definitely the most important. Okay, so maintaining homeostasis. You got to be. How does it do that? Well, it's going to do it by this is pretty much your central nervous system, your brains, your nerves, all that is going to control your surroundings. Okay, in order to do that, you need energy. So this is going to be another characteristic there. Another one as well. All animals are going to be able to do this. So we'll talk about this. We'll talk about some generalities with their different animal types. This is just a general introduction to how animals survive. So this now is 25.2. Okay, so this is going to be what separates and classifies all the different animal types, okay, between invertebrates and vertebrates. And you have different features that you are going to need to understand. Now, I will supply a chart with everything filled out, but you will be answering the question for our daily question of what they are and a brief definition to these. So there's 7 or 8 on them, depending on if you want to break some of these up. Some of these can be put into multiple columns or categories, but we'll go ahead and list 7 here. So first I want to talk about level of organization. Okay, going back to way back last chapter, here is that level of organization. Okay, and starting from your molecular, or what's even smaller than that, atoms make up molecules. Molecules then are going to be cells. Okay, cells, then a collection of cells going to make up tissues. Tissues make up organs, organ systems, and so forth. So if we're looking at it, and as evolution would put it, or the textbooks would say, evolution is that the earliest animals are going to be most simple. And then these organs and complex animals are going to develop. Okay, so looking at this, you have here the level of organization, and then you have a kind of a picture diagram. I'm not going to get into too much detail. Just know that if you do not possess as much of these up here, you are less complex. You're more simple if you're just a cell. Right, just a cell is the most simple organism you can have. So an amoeba, things like that, singular cellular animals. Tissues are going to be more complex, and organ systems and things like that. So going on to body symmetry. Symmetry is being, if you kind of put a mirror in it, it's going to reflect. Now we are created with symmetry. Okay, and that's part of God's creation. If you look into it, we can break that down into even more as far as different mathematical equation. I don't want to mispronounce the guy's name, but there's certain numbers and things like that are repeated over and over and over in nature, and this is what causes this symmetry. So there are going to be 2 different types of symmetry. Radial symmetry and bilateral symmetry. So this first one here, radial symmetry, if we're looking at this, think of it as kind of a tire. Okay, that is a terrible tire, but it will work. And a radial symmetry is you're going to have this kind of axis in the middle, and you're going to be able to cut it up many different ways. So your circular kind of animals are going to have this. All right. So here you see an enemy here that is going to be able to get cut into many different ways. And then you have the next one, which is a bilateral symmetry. And this is a single plane. So I'm going to draw kind of an example of a bilateral symmetry. So this is a person. This is not all that great, but if you draw a line straight through you, this line is actually called something on your body. It's called your midline. So humans have what is called a bilateral. Bi meaning 2. So bilateral is you're going to split that organism into 2 equal, left or right, top or bottom. Doesn't necessarily have to be left or right, but that's most likely that, and it's going to be symmetrical for the most part. Okay, so if you look here at this lobster, there's a plane there that's going to split it, and you're going to have a left here, and you're going to have a right there. Okay, so now these germ layers here, I want to talk about that. So, okay, so overall, that's the first. This is your first body plan, body symmetry, okay, and that's just purely visible. You can look at it, be able to, divide it up. Now you have germ layers, okay, so these germ layers, germs are going to, when things germinate, when we talk about plants and you have the germination, right, that's when things are growing. So this is going to be starting to talk a little bit about into embryology and things like that. So how a organism is going to develop from zygote to maturity. Okay, so there's 3 layers here, and these layers are going to cause a different function. Okay, as they develop. Okay, so here you have the endoderm, endo being inside, okay, has an in here, so being inside, okay, and it's the innermost layer. Okay, then you have meso being the middle, all right, or uh, middle there, tissue layer, and then ecto being the outside. Okay, so you have these three layers that are going to help develop your body differently. Okay, endoderm being on the inside. If we're looking at a human, you're going to have a little bit more developed of like the digestive system, respiratory tract, things like that. Things that are going to be kind of inside your body. And then here, mesoderm being your muscles, organs, things like that, your cavities that are going to form. And then ectoderm being the outside or outer tissue. Okay, so this is going to be your two, or your second is going to be a little bit about these germ layers. Okay, so let's carry on and talk about how body cavities are going to form, and it's different. This is going to help develop differences between animals. Okay, these are called coelums. Okay, so if you have a category or a cavity, you're going to be called a coelom. Okay, it's not coelom, it's called a coelom. I don't know why. Okay, but a coelom, a body cavity that develops within the mesoderm and is completely lined with tissue derived from mesoderm. Okay, so if we're looking at our tissues again, remember, you have ectoderm being on the outside. That's going to be this blue. Okay, you have mesoderm being the pink, kind of like muscle right in the middle there. And then you have endoderm here being on the inside here. So now you have this opening here, and that's going to be the digestive cavity. Okay, in your body, we're going to look something similar to actually what's over here, and you're going to have organs and things here. Okay, but here you have no cavity, right? There's no room in here. There's no cavities there to house organs and things like that. So it's going to be a very simple animal. Okay, and then you have stuff here. There is cavity here. What is a pseudocoelomate? Okay, meaning it's false. It's not connected. Right. The reason why is yes, there's a cavity, but there's no connection between this the ectoderm and the endoderm. Okay, there's just this gap here. These are going to be animals that their circulatory system is not closed. They just kind of open and use gravity and force to push through it. Okay, these aren't going to have very complex structures. These aren't either. Now, coelomates or coeloms are going to allow for organs to develop. Right now, there's space here. You have the mesoderm here. Okay, that's connected, and now you have space here for these organs and very more complex organisms. Okay, humans, we are what are coelomates. Okay, so that being the third body plan. Okay, now you have embryological development. Okay, so what we're talking about here is kind of this blastula. We need to know what a blastula is. Okay, this is a very important word because this is what we are talking about as far as the difference here. So zygote to blastula, that's all the same. Okay, zygote, that's a blastula here. And what it is kind of this hollow ball. If you got a hollow type of chocolate or egg or something like that for Easter, that's what this looks like. It's just hollow in the middle, and based on your DNA and things like that, it's going to determine how you are going to develop. So it's not actually developing inside out. It's if you look here, it's going to develop outside in, and that's what's going to happen here. Okay, and what's going to form here is it's going to connect here, and it's going to try and form what is the digestive system. Okay, or the digestive canal, I should say. So that's why you see the gastrula, that's that canal, or that same thing, kind of hollow ball, but it's going to be now your digestive system. Okay, so there's two types. All right, there's what is called a protostome and a deuterostome. Okay, a protostome blastula is first going to form the mouth. Okay, so this here, if this was a protostome, this is going to be the mouth opening. Okay, and deuterostomes are first going to form the anus. Okay, so that's just as far as their stages of development. Okay, you have those being the different parts that are going to develop first. Okay, but you can see here that you're going to develop the digestive system first. To be honest, I'm really not sure what number we're on, but I'm pretty sure we're on 4. We'll see if that works at the end. So here we go. You can see this kind of, here you go, 8 cell, and then you got to get some breaking down. This one here adds your germ layers here, so you can kind of see the germ layers forming a little bit. Now if you're looking at protostomes, this will be the anus, this will be the mouth. Here you have the mesoderm being in here. Okay, so the mesoderm there, and then you have organs, things that will form as well. And then you have deuterostome. So mouth, anus, it's just different. Okay, so one forms into the other first, but both get made. It's just one before the other, I guess. So looking at it, here is your germ layers and cavities and things like that. So looking at it, I guess I'll write on here as far as the body plan. Okay, these are your cavities, which I believe were 3. Okay, and then this is your embryological development. Okay, so 3, 4. Here's a cross section. So this cross section, this plane here is what that scalpel of the knife being. Okay, it's going to show. All right, and this is what it would be through a coelom, or a coelomate, pseudocoelomate, and a acoelomate. Okay, so it's kind of squished. You see, this is a flat worm here. Okay, this is a round worm, maybe an annelid here. It looks more like an annelid, and you will understand. Oh, it's a nematode. Okay, so here you'll have a nematode, which you see here. Once again, opening space here, but it's not connected. And you can see here, during development, to build and make all these organisms, okay, or these organs that make more complex, you need a mesoderm that's connected everything. You see here that mesoderm is going to be connected everywhere. So in this process here, that mesoderm is not going to connect on the inside here. Okay, this yellow being the endoderm, it's not connected, and there's no space really for it to grow. Cladograms. Okay, cladograms, you're going to be looking at these. These are the different types of animals as far as classification of animals that we're really going to look at. Basics, okay, we'll get into more so when we start talking about animal systems and things like that. We'll split more into, we're going to be looking more into the chordates. We'll talk a little bit about stuff here, but these are very, very simple animals. Okay, so if we're looking at the levels of complexity, you're going from simple. Sorry, this is make way. From simple to complex. Okay, and we should know what a cladogram is, as we already used some. Okay, so you're going to see here all these kind of different types of body plans that are here. So you have symmetry. Okay, you have deuterostome, you have pseudocoeloms, you have coelomates, you have all these different things here. So sponges being the least complex or most simple. So that's what this is saying, is that you came from a sponge. If that's what your textbook were to be true. So think about it. Go grab that sponge that you wash your dishes with and think that your textbooks are teaching you that you came from that. Yeah, weird. Okay. So the cladogram is this science just trying to put together how these things are related and what came first and what not. I think of it more of a way as trying to classify these different animals. What makes an annelid different than a mollusk or a roundworm, right? This is a worm. Okay, what makes it different than this worm and that worm, right? You have 3 different types of worm. Why is that worm different than this worm? Well, like I said, God makes everything a certain way, right? We are made in His image. So therefore, these are made in His image as well, and these are different. So science is just trying to put together why, okay? And finally, I believe this is our last slide for the most part. This is that table that I wanted to talk to you about. Okay, these are your different levels of your body plans, but you have all the different animals here. Okay, so you have the germ layers here. Okay, so we look at this and kind of label this. That's 1, 2, 3, 4, 5, 6, 7, 8. Okay, so those are your 8 levels of or body plan. Okay, now all animals, not all animals are going to have all 8 of these. Some may be absent. Okay, and that's what that absent means. Okay, body cavities. Here, this means that it's not plausible. Okay, so therefore, you only have 3 really that are going to 3 sections. But for the most part, out of the different animals, there's 9 different types of animals. Okay, all these body plans. Now you need to know the difference between these. Your quiz, for the most part, I'm going to be asking you a lot about this and using this. Okay, so your question for the day today, you're pretty much just explaining what these are. Okay, you're going to use it, list it, and then explain it. Okay, so a sponge, you can see here, specialized cells. It only has cells. It doesn't have organelles, it doesn't have organs or organ systems. It's cells. Okay, so all the cells are going to produce what is needed in order for it to survive. Okay, and we'll talk about a lot more of the systems and things like that. But no body symmetry, no germ layers, no segmentation. Segmentations are going to be repeated segments. Okay, which I'll get to when I get to chordates. But that is going to be what. Also, cephalization. This is going to be a nervous system for the most part, some sort of nervous system. Okay, so segmentation are going to be segments. Okay, you can also throw in here appendages. Okay, appendages as well for segments. So very basic, like I said, sponges being the most basic out of everything, and they only have cells. Now we make our way here, right? So if we go in left to right, we are going to more complex. Well, what's the difference? Well, now you start to have tissues. Now you have symmetry. Okay, these here sponges are the only asymmetrical organism. Okay, or I shouldn't say asymmetrical, but or organism. They're asymmetrical animal. All right, they do not have body symmetry. They have no germ layers. This has 2 germ layers, so it doesn't have all 3, and that's what is represented here. Okay, it is an acoelom, meaning there's no cavities here. All right, and then everything else is separate, or sorry, absent. Then you have arthropods. Okay, these are your insects. This is the most diverse group of animals there is. I do not remember what the number is off the top of my head, but millions of different types of arthropods. Okay, so arthropods are going to be your insects, your grasshoppers, things like that. Okay, so now you have organelles, or sorry, organs. You have bilateral symmetry. You have 3 germ layers. You have a coelom. You have protostome. Okay, and here you're going to have segmentation and cephalization. Okay, so they have some sort of nerve. Most of them are going to use what is a nerve net with a little bit of nervous tissue called their brain, or if you want to put it. Okay, then you have roundworms. Okay, these are your, I don't know any off the top of my head. Anyways, I can't think of any as far as specific animals that are called roundworms. I want to say a tapeworm is a roundworm, but don't quote me on that. Bilateral symmetry, 3 germ layers. Now we're looking into kind of differences here. Bilateral, 3 here, organs. So now what makes a roundworm different than a flatworm, different than an annelid? These are your 3 worm types. Okay, so we look at it. Well, body cavity for a roundworm is a pseudocoelom, right? You have an acoelom here, and you have a true coelom. So they are different. Okay, look, if you look here, the embryological development of your annelids, protostome, 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 absent, absent, present, present, present. So the only thing that's different between an annelid, a roundworm, and a flatworm is the embryological, or sorry, the body cavities. Whether it's a pseudocoelomate, a acoelomate, or a coelomate. Okay, so you have 3 different classifications of worms, all with different body cavity types. Then you have mollusks. These are being your mostly your shellfish. Okay, with a couple of different exceptions. Okay, snails are part of your shellfish. Okay, or mollusks, I should say. You can see the kind of the snail there, but you're going to have other things such as crabs and things like that that are going to be mollusks as well. Bilateral, 3, true coelomate, protostome, absent, present. So once you get past cnidarians, that's a silent C there, so it's cnidarians. Once you get past that, you're just looking into, okay, what is the difference here? Okay, why is a mollusk going to be by itself? Okay, where you have, if we look at this, what's different between these? Okay, well, these are all the same, okay. But what's different is going to be the absence of segmentation, okay. So a mollusk is not going to have segmentation. All right, so you're going to have your snails and oysters and things like that. Your, like I said, shellfish, but not necessarily all shellfish are going to be in here, as you have appendages on like crabs and things like that, which I believe are going to be in your arthropods, as they are going to be part of your arachnopods. Anyways, going in, but here's the difference. Okay, so going in here, you have differences between your worms, in the body cavity. Then you have difference in segmentation between mollusks and annelids. Okay, echinoderms, well, you're going to have a big difference here being radial. Okay, it is one of the only radials, that other being the cnidarians, or your jellyfish. Okay, so you have jellyfishes here, starfish, snails. These are going to be your earthworms. Flatworms are going to be those kind of sea worms that kind of look like ribbon floating through the water. Roundworms, nematodes, I want to say, they're going to be slick, okay, smooth, also called smooth worms. Arthropods is your insects, sponges. Echinoderms being your starfish, and then chordates everything else. Okay, so everything that you can think of, all your pets, everything like that. Now, what's the difference between this? Okay, well, bilateral, but we have what is a deuterostome. Okay, so pretty much your quiz is I'm going to ask you, give you kind of a rundown of this, and you're going to have to tell me what classification of animal. All right, so if I tell you what animal has organs, bilateral symmetry, germ layers, pseudocoelomate, protostome absent, or protostome segmentation is absent, encephalization is present, well, you just use this chart and you can find it. So you're just kind of checking it off as you go. So that's chapter 25. Okay, thanks for coming back, and best of luck on your quiz. And please let me know if you have any questions. Email me or write them on Google Class. Have a blessed day.